Good morning. Today's topic is hemoglobinopathies and sickle hemoglobin. The learning objectives of today's topic are: describe and discuss hemoglobinopathies, describe and discuss sickle hemoglobin, then hemoglobinopathies. Introduction to hemoglobinopathies was given in the last lecture, and today we are going to learn it in detail. So hemoglobinopathies. Hemoglobinopathies are a group of disorder due to alteration in hemoglobin structure or impaired synthesis of polypeptide chains. Hemoglobinopathies may be congenital or acquired. So there are 4 different types of hemoglobinopathies. So one is the hemoglobinopathy due to the qualitative change, that is change in the structure, and second one is the hemoglobinopathies due to impaired synthesis of polypeptide chain, that is quantitative hemoglobinopathy, and the third one is hemoglobinopathy may be congenital, so that is may be inherited, and the fourth one is acquired. So the hemoglobinopathy may be acquired due to toxic exposure to the different substances. So 4 different types of hemoglobinopathies. So hemoglobinopathy may be due to the alteration in the hemoglobin structure, that is qualitative change, may be due to the quantitative change, or it may be due to the congenital, so that it may be inherited directly from the ancestors, or it may be acquired due to the toxic exposure. So 4 different types. Let us see them one by one in detail. So here is the flowchart or classification, so where you can see the different types of hemoglobinopathies. So congenital, so it is the inherited disorder of the hemoglobinopathies. Acquired is the disorder of the hemoglobinopathy which has been acquired due to the toxic exposure of different chemicals or substances in the lifespan of a person. So congenital hemoglobinopathies is of again 2 types, that is qualitative and quantitative change. So the qualitative, under the qualitative change there comes the structural hemoglobinopathies, which is associated with altered amino acid sequence of a globin chain due to mutation. So it may be a point mutation. The examples for this is HbS, HbC and HbE. Then the second one is thalassemia. So thalassemia is an example of congenital hemoglobinopathy which is related to a quantitative change. So where there is defective biosynthesis of the globin chain. The example for this is AlphaThalassemia and beta-thalassemia. Depending on the defects in the chain. If it is associated with the defect in the alpha chain, it is alpha-thalassemia. If it is associated with the defect in the beta chain, it is beta-thalassemia. Then the next one is acquired. So acquired is hemoglobinopathy is a type of hemoglobinopathy which is acquired during the lifespan of an individual due to the toxic exposure to the different chemicals or substances. So the example for this acquired hemoglobinopathies are Methamoglobin, sulfahemoglobin and CarboxyHemoglobin. So today we are going to learn in detail about them all. So let us see one by one. Then abnormal hemoglobin. Mutation in the gene that code for globin chain, that is alpha, beta, gamma and delta, can affect their formation and biological function of hemoglobin. Such hemoglobins are called as abnormal hemoglobin. When biological function is altered due to a mutation in hemoglobin, the condition is called as hemoglobinopathy. And thalassemia. So it is a quantitative change of hemoglobinopathy. So thalassemia is a group of genetically transmitted disorders of hemoglobin synthesis due to the lack or decreased synthesis of alpha or beta globin chains. So a quantitative change. So this type of hemoglobinopathy is referred to congenital quantitative change. So it is inherited, and the change here is quantitative, not qualitative. So thalassemia is a group of genetically transmitted disorder of hemoglobin synthesis due to the lack or decreased synthesis of alpha or beta globin chains. Because the synthesis of one globin chain is reduced, there is a relative excess synthesis of other globin chain. These globin chains may precipitate in the cell, causing hemolysis, resulting in hypochromic anemia. The name of this group of disease comes from the Greek word thalassa, meaning sea. Because this disorder occurs more commonly among the people living near the Mediterranean Sea, hence it is called as thalassemia. Types of thalassemia. Depending upon whether the genetic defect lies in the synthesis of alpha or beta globin chains, the thalassemia is classified into alpha thalassemia or beta thalassemia respectively. If the defect is associated with the alpha chain, it is called as alpha thalassemia, and if the genetic defect of synthesis is present, is associated with beta chain, then it is called as beta thalassemia. Alpha thalassemia. Synthesis of alpha globin chain is defective here. The alpha globin chains are coded by 4 copies of alpha globin genes. The alpha thalassemia results from the genetic defect in one or more copies of alpha globin genes and is characterized by either decreased or total absence of synthesis of alpha globin chains. So depending on this, the alpha thalassemia is of 4 different types. So let us see them one by one. So the first one is silent carrier type of alpha thalassemia. So here only one of the 4 copies of alpha globin genes are mutated. The remaining 3 are normal. So they do not show any clinical symptoms of thalassemia as the other 3 genes are normal. So then the second one, that is alpha thalassemia trait. Here out of the 4, the 2 of the 4 copies of alpha globin genes are mutated. So they usually have only mild anemia and is not a fatal disorder. Then the third one is hemoglobin Hedge disease. So here out of the 4 copies, the 3 of the alpha globin genes are mutated. They have moderately severe hemolytic anemia. Then the last one, that is hydrops fetalis. Here all the 4 copies of alpha globin genes are mutated. So this is a very lethal condition. Most of the affected are stillborn. Stillborn means the death before the birth, or they may die soon after birth. So it is the most lethal of all the alpha thalassemias. Then beta thalassemia. Synthesis of beta globin chain is impaired due to genetic defect in beta globin genes. So beta globin genes are coded by 2 copies of beta globin genes and is characterized by decrease or total absence of synthesis of beta globin chains. So beta thalassemia is of 2 types. That is beta thalassemia minor and beta thalassemia major. So alpha thalassemia is 4 types, but beta thalassemia is of 2 types, because here so beta chains are coded by 2 copies of beta globin genes. That's why it is of 2 types, that is beta thalassemia minor and beta thalassemia major. So let us learn about them. So beta thalassemia minor is also known as beta thalassemia trait. Here one of the 2 copies of beta globin genes is mutated. It is a heterozygous state. The presence of one normal gene in the heterozygous state allows enough normal globin chain synthesis so that the affected individuals are usually asymptomatic. The individual may be completely normal or has a mild anemia. Then the second one, that is beta thalassemia major. The beta thalassemia major is a homozygous state carrying both the genes. So 2 mutated beta globin genes. So it has 2 and both the 2 are mutated. Thalassemia major leads to severe anemia. They regularly need blood transfusion. Bone marrow transplant has recently been introduced as a remedy for beta thalassemia major. Then sickle anemia and sickle hemoglobin. So what is the sickle anemia? So sickle cell anemia is a genetic disorder caused by production of an abnormal hemoglobin known as sickle hemoglobin. So here it is a qualitative change. Thalassemia is a quantitative change, and now you are going to learn about the qualitative change in hemoglobin structure. So sickle anemia is an example of a qualitative change. So sickle cell anemia is a genetic disorder caused by production of an abnormal hemoglobin known as sickle hemoglobin. The production of hemoglobin, yes, is due to mutation in the beta globin gene, which codes for beta globin chain. The mutant beta globin chain of HbS has an altered amino acid sequence. So here in HbS, the glutamic acid residues normally present in the sixth position in the normal healthy adult hemoglobin. But in the HbS individual, the beta chain of HbA is replaced by valine residues as a result of mutation in the beta globin chain. So in the normal healthy adults, the beta chain, the sixth position, the glutamic acid is present. But in HbS, the individual who contains HbS or sickle cell anemia, individuals with sickle cell anemia, there is a mutation in this beta chain at sixth position where the glutamic acid is replaced by valine. So it is a good example for point mutation. So where an amino acid, one amino acid is replaced by another amino acid. So here all the 570, so hemoglobin is made up of 574 amino acids, where the 573 amino acids are normal, but the difference is in only one amino acid which is present at sixth position. So even though the 573 amino acids are normal, due to one abnormal amino acid, so the life of the person has been changed. So the lifespan of RBC is also changed. So the normal lifespan of RBC is 120 days, but the lifespan of sickle cell RBC is around 17 days. So such a gigantic change is observed due to this point mutation of sickle cell anemia at sixth position of the beta globin chain, where the glutamic acid should be present in the normal healthy adults, which is replaced due to the point mutation by valine. So let us see what happens when glutamic acid is replaced by valine in sickle cell anemia. As the polar or hydrophilic glutamic acid residue is replaced by non-polar hydrophobic valine, it generates hydrophobic contact points called sticky patch on the surface of beta globin chain. So this alteration markedly reduces the solubility of deoxygenated sickle cell hemoglobin. So the solubility decreases. When the HbS is deoxygenated, the sticky patches can bind to one another deoxygenated HbS molecule. This binding causes polymerization of deoxy-HbS, forming insoluble long tubular fiber of HbS. The insoluble fiber of deoxygenated HbS are responsible for deforming the red blood cells, which looks like a blade of sickle. Hence the name of the disease is sickle cell anemia. So here is the structure. So here is the normal adult hemoglobin, and here is the sickle hemoglobin, where due to the mutation, the blade-like structure is formed on the beta globin chain, and which is responsible for polymerization of deoxy sickle cell hemoglobin. So here it is the polymerization, the schematic representation of polymerization of deoxyhemoglobin S molecules and formation of tubular fibrous structures. So due to the formation of this tubular fibrous structure, the red blood cell appears in the form of sickle. So sickle red blood cell. So sickled red blood cells lose water, become fragile, and have a much shorter lifespan than the normal cells. So if the normal adult hemoglobin lifespan is 120 days, the lifespan of the sickle red blood cell is around 70 days, leading to lysis of the red blood cells and results in hemolytic anemia called sickle cell anemia. The more serious consequence is that small blood capillaries in different organs becomes blocked by long abnormally shaped red cells. This interrupts the supply of oxygen and leads to anoxia, that is oxygen deprivation, which causes pain and eventually death of the cell. So sickle cell anemia is of two different types. It may be homozygous or heterozygous. So let us learn about it. So sickle cell anemia is a homozygous disorder in which the individual has inherited two mutant globin genes, one from each parent. So it is characterized by chronic hemolytic anemia, tissue damage and pain, and increased susceptibility to the infection. Such patients usually die in their adult age. Then sickle cell trait is a heterozygous trait. So sickle cell anemia is a homozygous trait and sickle cell trait is a heterozygous state in which the individuals have received the abnormal mutated beta globin gene from only one parent and have one normal gene. So they do not show any clinical symptoms and have normal lifespan. Persons with sickle cell trait are resistant to malaria caused by Plasmodium falciparum. This parasite spends an obligatory part of its life cycle in the red blood cells. Since the sickle red blood cells have a shorter lifespan than the normal red blood cell, the parasite cannot complete its life cycle. Hence, they are resistant for malaria. So, sickle cell trait persons are resistant to the malaria and they do not show any clinical symptoms and have a normal lifespan. Then HbC or Cooley's hemoglobin. So HbC, here it is also a qualitative change, example for qualitative change. So here the glutamic acid at position 6 in the beta chain is replaced by lysine. In HbS, the same glutamic acid at 6th position is replaced by valine. So instead of valine in HbC, this glutamic acid is replaced by lysine. So it becomes HbC if lysine, and it becomes sickle cell anemia if it is valine. So both are the good examples of point mutations. The red blood cells of people with HbC do not sickle. However, crystals of HbC may form within the cells. Both homozygous and heterozygous individuals of the disease are known. So this disease is characterized by mild hemolytic anemia in homozygous individuals, and clinically the heterozygous individuals are asymptomatic. So HbC, where the glutamic acid at 6th position of the beta chain is replaced by lysine. And HbM, so mutation in histidine residue of either alpha or beta chain which bound with iron in the heme molecule. In HbM, the histidine is replaced by tyrosine and the iron is stabilized in ferric form instead of ferrous form, which cannot bind with oxygen and leads to cyanosis. The letter M of HbM signifies that the affected chain are in Methamoglobin form. So due to the mutation here, the histidine is replaced by tyrosine. And due to this mutation, the iron is stabilized in ferric form instead of ferrous form. So due to this ferric form, the oxygen cannot bind to it. Instead of oxygen, the water molecule binds to it and it leads to cyanosis. So here you can see in the diagram about HbM, where the proximal histidine is substituted by tyrosine, results in the formation of HbM, and in the HbM, the water rather than oxygen is bound at 6th coordination point in HbM, which leads to cyanosis. And abnormal hemoglobin derivatives, they are methemoglobin and carboxyhemoglobin. So methemoglobin, the iron normally present in heme is in ferrous state, which is being replaced by ferric state in methemoglobin. The ferrous of hemoglobin is oxidized to ferric state by superoxide by certain drugs or chemicals or other oxidizing agents, forming methemoglobin, which cannot transport oxygen. Only a very small amount of methemoglobin is present in the normal blood, less than 1% of the total hemoglobin, formed by spontaneous oxidation of hemoglobin. Then carboxyhemoglobin. Carbon monoxide combines with heme moiety in hemoglobin. It combines at same position in the hemoglobin molecule as oxygen, but with an affinity about 210 times greater than oxygen. As a result of this, even small quantity of carbon monoxide in the inspired air causes the formation of relatively large amount of carboxyhemoglobin with a corresponding reduction in oxygen carrying capacity of the blood. Even as little as 1% of carbon monoxide in the inspired air can be fatal in minutes. So it is very fatal. So this completes your lecture today, and this is the assignment which you need to complete it and send it back to me on Telegram. Thank you.